Next up, we have a tag team talk on first steps toward large-scale experimental benchmarking of Rosetta design protocol performance. Take it away, guys.
All right, thanks, Doug. Okay, so first off, this is actually a very new project. We're just very excited about it, so we wanted to share right away the things that we're doing and what we're thinking about, partly because we want to solicit feedback from everybody on the things that we should do, also to let people know about what we're doing. We want this, ultimately, our project today and the themes we want to be something that becomes a community-wide resource, and so we want to engage the community as early as possible. So really the question we're trying to ask is, how do we improve the fidelity of our designs, right? I mean, Nihal gave a great talk where she tested tens of thousands of things and got a small number. But I think ultimately what we want to do is we dream of a day where you order one design and that works, right? But I think in order to get there, we need to start systematically evaluating what we're doing when we're doing design. Right? We need to empirically assess our efficacy. There's a lot of knobs in Rosetta, and if you dial a little bit, does that make your design protocol more likely to produce successful designs or less likely? Where exactly do these knobs need to be? And kind of right now, the state of the game is you kind of use a lot of intuition. You write a protocol, and then you test 100 designs, 20,000 designs, whatever. If one works, great. You get your high impact paper and you move on. And we don't really spend time evaluating like how do we make this more reliable in a systematic way. And so that's what we really want to do. We want to come up with a way that we can compare our enhancements to the algorithms to the current state of the art and really see that we're making stuff better and quantitatively measure that. And I think this kind of fundamentally represents a paradigm shift in how we develop Rosetta. And I think that if we all really start thinking about doing this, that everyone will benefit and we'll find that Rosetta design gets, a 5% improvement in fidelity is great. Maybe not revolutionary on its own, but we start stacking those 5% improvements. Suddenly, we may find ourselves living in a world where you can depend on the output of Rosetta to actually work that way. Okay, so we need to benchmark with lots of different protein structures to assess overall performance. We don't want to design one protein, right? We want to design protein as a general class of material. And so high throughput experimentation is going to be required to do this. And so that is also a little bit about talking about what sort of high throughput experimentation we're talking about. Okay, so overall goal of this study is to perform a large-scale experimental assessment of energy functions and also design algorithms. And we want to empirically determine which is the most effective. So our overall outline, you probably have seen or heard about Jack's talk from summer RosettaCON. So you discovered a problem with fast design. It's something that a lot of us have faced over the years, and there's lots of little hacky workarounds. Well, Jack has figured out really what the problem was, so he'll talk about that in detail. Then we'll briefly describe how we can test the efficacy of a design protocol, and then I'll share with you the experimental voodoo that we use to make this happen. So pass the baton.
All right, thank you. So, like Chris mentioned, I had a short talk on this over the summer that some of you were at, but I'm going to summarize the takeaway from that and just walk through a little bit about, walk through what fast design does when it tries to design a protein. So here I'm handing fast design. It could be an interface monomer, but I'm trying to just show a protein. The first thing fast design does is it repacks designing with very soft repulsion, 2% of the final repulsive weight. And this gives you enormous side chains that would normally be considered clashes, but the score function is so soft that that's not an issue. The next step is then minimization, still at 2% softness. And even with the large side chains, the backbone collapses towards itself. Again, interface or monomer core. These two steps are repeated with increasing weights. So 25%, then 55% until it's back to 100%. But every time that it moves up a step in repulsion, it's always the the packer that sees this new increase in clashing, and the only thing you can do to get rid of those clashes is to just design away the side chains down to alanine. And then it's the minimizer's turn, and there's no clashes, so the backbone stays collapsed in. And that just propagates, and then you end up at the end of fast design with a smaller protein than you started with and a lot of alanines. So in the months since then, we've been trying to address this, and I'll summarize it here without getting into too much detail, but we focused on changing the ramping pattern for the repulsive weights, not just increasing them, but also increasing them between packing and minimization so that the both protocols can handle clashes as you remove softness. I tried many different design cases: relaxed crystals, generated decoys from ab initio and sewing, monomers, interfaces. I've done design, relaxed with no design. You actually still get a little bit of this problem when you can't even design combinations, so like one-sided interface design. So I tried to cover a lot of different cases and not optimize for one. And luckily, most of them agreed in what direction was better. But we were able to increase repulsion in a way that improved all of these. We optimized for score and structure retention, so alanine concentration and the radius of gyration of the protein compared to when you start fast design. Luckily, these all correlated together, so we never really had to choose one over the other. They all agreed what was better. But this isn't enough. We found that just increasing repulsion gave us funky amino acid distributions. I think this might be small. So what I'm plotting here is the amino acid distributions for the native in blue. This is for relaxed crystal interface one-sided design, but it's similar for all cases. So the native distribution in blue, fast design as we all use it with the default behavior in orange. And then what I just talked about, increased repulsion in gray. And then what I'm going to end up with is it's named KILA 2019. It kills the alanine distribution. After seeing Franklin 2019, named after women in science, it seems kind of silly to have this. And on that note, the credit for naming goes to Andrew Lee Burkray. I'm going to zoom in.
Something after Shannon Thornton.
Sorry.
I suggest something for Shannon Thornton.
And this is just zoomed in. Same data. I figured the previous plot would be too small, but points of interest. What KILA does is it not only ramps the repulsive weights that we get from the increased repulsion, but it refits the reference values for each repulsive weight for each individual score function. So even when you're packing with a very soft score function, you still can expect to have a normal amino acid distribution. And this corrects some things, so like isoleucine and leucine, tryptophan. When you increase repulsion, you actually get a ton of those, and you lose a lot of serines, polars. And so in green, we're returning almost all the way back to native. I'll go through this quickly, but this is in master. You can use it if you're interested. If you're not interested in the reference ramping, you can use the case-specific monomer design, monomer relax, and so on the right. This is online. You can find it by Googling Rosetta KILA 2019, but there's a link, and the slides are posted on the wiki, so you can go there. Also, there's no reason to use the RosettaCON 2018 script that I posted over the summer to give people it, so that's deprecated.
Great. Cool. All right. So talk a little bit now about how we think about testing this, right? So we've got these great protocol improvements, but we want to see, people have been using fast design for years now, sort of in the old-fashioned broken way with hacks, right? But we want to quantify, like, how much better is this new thing? How do we actually go about testing this, right? So we need to make a fundamental assumption. And so our first assumption, which may or may not be true, is that better Rosetta energy will mean better thermostability. In theory, I shouldn't say in theory, well, maybe we'll have demonstrated that the Rosetta energy correlates with a delta G of folding that we now put in kcals per mole. So if a protein is in a deeper energy well, has a better Rosetta energy score, should take more input energy to kick that out of the energy well. And so we would assume that thermostability should correlate with Rosetta energy. So this is our fundamental assumption that we're working on. If people have other ideas for other assumptions that we could test, I would love to talk to you. So we also need a library of native protein structures that we can use to test this assumption on. So Frank DeMeo is kind enough to provide a library of high-resolution native monomer structures that he uses to train the energy function. Mini proteins are not the same thing as what we call like regular globular proteins, right? You can do a lot of high-throughput stuff with mini proteins, and Gabe Roslin has done some very exciting stuff with that. And if he were here, I know he would agree with me, saying that all the rules for folding a mini protein probably cannot tell you all the rules for folding regular-sized globular proteins. So we need to work up a little bit in size and scale of proteins. So the sort of traditional high-throughput service display methods might not necessarily cut it for this. So really what we're going to do is we're going to take a library of native structures, we're going to redesign them with the different fast design protocols, we're going to test different energy functions, then we're going to individually produce and purify and biochemically characterize each one of these one at a time. And we can reach throughputs of about 10 to the 3. So it'd be really nice to be able to get to, say, 10 to the 5. But when you reach those types of throughput, you end up having lots of measurements that are kind of low quality that you have to make a lot of inferences with. In this case, we can start to reach, we can do 10 to the 3, but with like really precise, high quality biochemical measurements. Okay, so we wanted to select a smaller set of natives that we would then redesign and test. So we wanted to narrow down to a manageable number to start off with. In some ways, this is really like a pilot experiment to make sure that we can actually do all this. And so we had quite a few criteria. We wanted to bias ourselves towards success. We wanted proteins that were previously expressed from E. coli. We want high resolution structures, so we know, or maybe not, Simon gave a great talk. We hope that the structures are accurate and correct. And so high resolution helps to ensure that that's the case. We want the structures to be complete. We don't want chain breaks where there'll be regions that we're redesigning that are not represented in the initial structure. We want these things to be bigger globular proteins. So we don't want anything smaller than 100 amino acids. We want them to be single domain and monomeric. So we don't have to worry about breaking protein interfaces. We don't want them to have disulfide bonds that can also throw off assessment of denaturation. Disulfides can stabilize proteins. We don't want ligands. We have to worry about interfering with ligand binding pockets. We also want the ability for our redesign to improve the protein thermal stability. So we don't want sequences from thermophiles or hyperthermophiles, and synthetic proteins also tend to be very thermophilic, so we don't want any of those. And we want it to be a unique structure. We don't want just to get a bunch of helical bundles or just a bunch of beta barrels. We want lots of different things. So we performed a pairwise superposition matrix, and no two proteins are similar to each other. You can have regions that are really similar, but if you have 100 main chain atoms, we don't want the RMSD to be any better than 5 angstrom over that stretch of structure. And then also it just finally went through kind of old-fashioned Rosetta, just like looked at these things by eye. You don't want stuff that's really funky, right? Want things that look like they could fold on their own, sort of Amundsen style, that you could theorize would actually fold up this way without a lot of help from chaperones.
Right. So we ran fast design with Killa 2019 on all of these monomers, and we really let it go. We didn't have any coordinate constraints or any constraints. Designed every position. And what I'm showing on the left is the Rosetta score per residue on the y-axis of the default fast design, and then fast design with Killa in orange. The x-axis is the score if you minimize the native. That's really more to separate the points than anything. Then on the right, it's the sequence similarity metric, which is the average Blossom 62 score of all the designable positions. So higher is better. Then the x-axis again is if you just don't make any mutations. What does the native give you? Just to separate the points. And you can see we get pretty good scores with Killa compared to what fast design used to give. And the sequences are more similar to native. So we're happy to see this.
Okay. So I'll wrap up and tell you about the high throughput way that we're actually testing all of these. So first off is we need to be able to move quickly. And so we spent quite a bit of time rethinking how we make proteins. How do we test lots of different things really quickly? And the first thing is we wanted to make it go faster between getting expression plasmid and getting some protein that you can test. And so if you've done sort of things the old-fashioned way, it normally takes 3 24-hour periods, which is really like 4 days, to actually go from DNA to a protein. So first you take your DNA, you transform it into bacteria, you plate it, you wait a night, right? Then you get your plate, you've got single clones, the colonies that have started from a single cell, so are clonal, you pick one of those, you put it into a new batch of media, you wait another night, right? Now you've got this starter culture, you take a little bit of those bacteria, you put it into a larger culture that you then grow a little bit more, you turn on expression, and you wait another night, right? So that process is very slow. Having to put it onto plates means that you require manual intervention of picking a colony and putting it into a new tube and transferring it. So there's a lot of steps that are not easily automatable, and it's very slow. So we wanted to make this go a lot faster. So we figured out a new way where you can transform your bacteria and then inoculate expression culture directly. So you skip 2 entire days, and now this is readily automatable. You don't have to do any like manual picking, things like that. And the method works really well. So we tried it with 2 different antibiotics. If you're curious as to why we did this, I'm happy to explain afterwards. And so the 3-day method here is in black. And so, oh boy. Great. Yeah, it's equivalent if you use carbonicillin as an antibiotic. And actually our method, our 1-day method, seems to improve protein yields if you use kanamycin. We're repeating this, we're not really sure why. And in this case, we're just using superfolder GFP, so we don't have to purify the protein to get our total yields. We're just looking, we're comparing everything. And so we're just using arbitrary fluorescence, like how much of this stuff gets made, and we can just put the bugs in a fluorescent plate reader and find out. Okay. So we're doing this in also autoinduction media. So a lot of times you grow up bugs and you have to wait for them to get to a specific density in the culture, and you turn on gene expression. So we have a formulated media that does this on its own. Also happy to talk about the genetic circuits involved in this later if people are interested. Another really big bottleneck in protein production is when you extract the soluble protein from the cells, right? You have to break open the cells, create a homogenate, and then you have this insoluble material that you have to separate away from your soluble protein. And that is inherently like a slow step that's resistant to automation. It usually involves spinning in a centrifuge, some sort of filtration that's very cloggable. So our big innovation here is to use magnetic affinity resin. And the nice thing about this is we can use detergents and enzymes to homogenize the cells and to break them open. And then we can take these magnetic beads, throw it right into the mess and the muck, and it will find our proteins that we care about out of all of it. And then we can put our resin on a magnet, and it will capture all the magnetic stuff, and we can aspirate all the junk and slime from the lysed cells, and we can resuspend the beads in fresh buffer and wash them to get particles off, and repeat this a few times so we can completely skip all clarification steps. So that also makes this really automatable. We can put this on a liquid handling robot and do this now, and it's much faster. And then finally, we use enzyme elution to release our protein of interest from the resin. So we use sumo tags. They help to facilitate folding, and there's a really great enzyme that we've spent quite a bit of time engineering that will cleave this. If people have questions about this, I'll also be happy to talk to you later about it. And the enzyme also is sequestered and captured into the resin. And the other really slow step is buffer exchange. So once you get your protein in a tube, in the traditional way, you have to put it in some sort of buffer that's compatible with whatever downstream biochemical assay you care about. Well, in this case, you can elute directly into your assay buffer. The protease is highly robust and doesn't seem to care what buffer it's working in. And so you can put it directly into your buffer so you can skip yet another step. If people are interested in learning this, we're putting together a paper that will detail all of this. And we're also working on putting together a boot camp. So people just came back from the PyRosetta and C++ boot camps. We would like to have a wet lab boot camp. So if you want to learn to do this, we can come and you can get a crash course for a week and make proteins in 24 hours. Okay, so what do these data look like? This is the initial screen of the 48 natives that we selected. You can see these little green dots indicate ones where we have a nice pure material. We're getting some uncleaved protein coming through. This is sort of an early stage. We still have a little bit of optimization. I think putting some excess beads in here can clean this up, but we're able to get about half of the native proteins. These are just like random sequences we grabbed from the PDB. So I think the ability to express and purify half of them totally at random is pretty nice. See, how are we doing on time, Doug?
Just give us 5 minutes.
Okay, go through this quickly. There weren't any really obvious features like why did this half work and the other half not work. Our average molecular weight is about 15 kilodaltons and overall, and the ones that worked, the same. Our distribution for topologies, we've got some that span the spectrum from helical to beta sheet and kind of everything in between. Most of our proteins are mixed alpha beta, and you can see that the overall in black here is the natives, but the ones that worked are in white, and it's basically just slightly smaller down, right? There's no particular, say, protein topology that's being enriched. So that's curious. And we can also measure the melting temperature and high throughput, so there's quite a few different ways to do this. This one we're using a dye that specifically binds to hydrophobic parts of protein, and so when a protein goes from a folded to unfolded transition with increasing temperature, the dye binds very rapidly at that transitional unfolding point, and you can use that to figure out where it melts. So you can see we span a wide range of melting temperatures with these natives from low 70s down to really like mid 30s. So there's lots of different representation across this, and our average is about 55. So we've got plenty of room for improvement. So this is quite nice as well. We don't have melting data for all of the 22. Some of them we didn't have yields that were high enough yet to actually determine those melting temperatures. Okay, so to wrap up, so we think that empirical assessment of design algorithms and different energy functions is going to bolster the modeling and design efforts of everybody. And we're quite excited about this and the potential it has to really improve everything that we do. So we have an automated high throughput way that we can make proteins and characterize them. And we plan to quantitatively assess how much like Killa 2019 improves the accuracy of our designs and the success rate. And we also plan to test the different energy functions head to head. Which one is really better for design? Will it be sort of our old friend score 12, or will it be the latest beta? So we'll actually empirically figure this out. So stay tuned. Hopefully by summer RosettaCON, we'll have the full set where we've redesigned these and tested, say, 500 designs and be able to tell you the best way to make your protein. So all the experimental work was done by one very talented research associate in my group, Ian Trubridge, and then also collaboration with Brian.
Yeah, so again, Andrew gets credit for naming Killa 2019, but also Andrew, David's here, and then Matt all provided structures for me to test Killa on, and hundreds of them, and it was very helpful. So, thank you.
Thanks, everyone.
We have time for one, maybe two quick questions. Good choice.
Oh, yeah, Rocco, go ahead.
Oh, I was just wondering, do you have a high throughput way of going from the sequence on the computer into the actual plasmid you're putting into the bacteria?
Oh, GenScript. We just order it in a plasmid already. Cloning is done by a company. Yeah, if you order in bulk, they give you good discounts. Yeah, Dan.
So if you have a list of magnetic beads that are being extracted around this, are you pulling down insoluble protein and bringing it straight to the lysate?
Right. So we do use nickel affinity. You're right, there's insoluble particles that come down in the initial mix, but when we wash the beads, we resuspend them. And so essentially they release any particles into the first, insoluble particles in the first wash, then we recapture the beads by putting it back onto the magnet. And so the particles sort of stay off of the beads. And after a couple washes, you can get that away. Plus, we're doing an enzyme elution. So normally when you elute from nickel, right, you're using imidazole, so it competes off everything that's binding. But in this case, we're specifically clipping off the one protein that's bound to the nickel.
Or I guess I mean, could you have proteins that end up in an inclusion body, but if a his tag is still exposed, you're pulling off a misfolded protein. You know what I mean? I don't know if you can pull it away from it. I don't know.
Right.
I'm just wondering.
Probably not. Inclusion bodies, I think, it's a in solution interaction, right? So if it's I don't know about that. Yeah, we try to stay away from insoluble stuff. It's hard to work with.
In the interest of time and staying on time, I think I'm going to cut you off.
Thanks very much.
Let's thank Jack and Chris one more time.